Under the umbrella of what are called immunotherapies are different types of therapy which include cytokines, vaccines, checkpoint inhibitors, and cell therapies. With cytokines, you're basically giving proteins that help immune cells grow, be healthy, and be more active so that they can then destroy the cancer. You can also administer a vaccine that works much as it would for an infectious disease, where you're trying to stimulate the immune cells present in the patient to make them more active and better able to see tumors. Checkpoint inhibitors are antibodies which allow immune cells to remain active and make them impervious to the tumor's attempts to shut them off. Or you can administer the actual immune cells to the patient. And these are the therapies that I'm personally most enthusiastic about and that our lab is heavily invested in. Now, there are different forms of cell therapy. Adoptive cell therapy using what are called tumor-infiltrating lymphocytes, or TILs, is a form of therapy in which patients get their tumors removed surgically. It's cut up into small pieces, and the immune cells that are present in the tumor, particularly what are called T cells, are grown in the lab until they reach multiple billions or even hundreds of billions of cells before being reinfused into the patient. And this helps eradicate any remaining tumor and protect from recurrence. But cell therapies also include immune cell engineering, where immune cells are modified by introducing a new gene that either allows the immune cells to function better or to see a tumor that was previously invisible to them. Now, among these forms of cell therapy, the best known is probably CAR T cells, which have been extremely impactful with liquid tumors such as lymphomas and myelomas. And these are immune cells that are engineered with what's called a chimeric antigen receptor, or CAR, to recognize proteins found on the surface of tumor cells, upon which they can destroy these cells. However, they haven't yet shown any success in solid tumors. Now, another option is TCR T cells, and these are cells that are engineered with a T cell receptor, or TCR. And both of these have strengths and weaknesses that make them relevant to different contexts. Now, cell therapies really only need one good target for them to be effective, and that means they could succeed in cases where tumors have an extremely low tumor mutational burden, like in the context of RET fusions. And this is an area that's been problematic for other immunotherapies, such as checkpoint inhibitors, because these tumors can be quite difficult for the immune system to see otherwise. However, a key limitation of cell therapies is that they must be personalized. The problem is that because the immune cells themselves are being administered as a treatment, there's a chance that the patient's own immune system can react against the treatment, which means they have to be engineered into each patient's own cells or personalized. This results in much greater costs and manufacturing challenges and is one of the reasons these therapies are further behind in the clinic. However, a key contrast to all other immunotherapies is that cell therapies are actually living therapies because those immune cells then remain in the patient and can protect them for the rest of their lives. So this is an area of great promise and an extremely active area of investigation for many labs around the world.